Hello, my name is Dr. Teresa Duda. For 30 years, I was a member of Dr. Sharma research group. Just as I remember, Dr. Sharma is a distinguished retired professor of biochemistry and molecular biology. Together with his research group and collaborators, he published 303 papers, including 20 monographs, 3 books, and he also held 2 US patents. He has organized and been a plenary speaker in 22 international symposia, amounting to the amount of $7.3 million. Dr. Sharma's work has been funded by numerous awarding agencies. First of all, National Institute of Health, National Science Foundation, Pennsylvania Lions Club, Eye Research Foundation, Damon Runyon-Walter Winchell Cancer Fund, and Veterans Administration. He is the discoverer of multi-limbed membrane guanylate cyclase molecule. Notably, without it, our universe would be lifeless and dark. It took Dr. Sharma 54 years, from 1969 to 2023, to accomplish this Herculean task. Only enormous stamina and perseverance allowed Dr. Sharma to conquer all obstacles which were set by influential and not always friendly peers. Dr. Sharma's life journey, personal and scientific, has been briefly narrated in sequential videos formats. The present video follows 3 previous. They narrated respectively topics of the life-sustaining molecule, video number 1, multi-limbed membrane guanylate cyclase, cellular signal transduction pathway at the molecular, genomic, and physiological level, video 2, and the evil saga that occurred between April 22, 1988 and August 6, 1993 to ruin Dr. Sharma's professional and personal life, video 3. The discovery of the multi-limbed membrane guanylate cyclase is fundamental in the field of cell biology. With a human touch, the present video and the next one will begin to narrate the history of the fundamental LIMP-1 adrenocortical hormone ACTH modulated membrane guanylate cyclase. Okay, so now let's talk to Dr. Sharma. Dr. Sharma, could you please elaborate why the discovery of the membrane guanylate cyclase was so important in understanding cell biology and signal transduction processes?
The discovery not only established the multi-limbed membrane guanylate cyclase signal transduction field, it also revolutionized the arts of developing clinical therapies to the diseases linked with the physiology of the endocrinology, cardiovasculature, sensory neurons, and cancer. Multi-limbed membrane guanylate cyclase molecule existed 250 million years ago. The internetted limbs throughout the central, sympathetic, and parasympathetic systems define their physiology. To do so, they generate common hormonal signal messenger, cyclic GMP, proving that cyclic GMP function as a cellular second messenger challenged the two dogmas existing at the time. First, there was a question of independent existence of this multi-limbed membrane guanylate cyclase pathway that was different from its previously known heme-dependent soluble form. Second, the only known hormonal cellular signaling pathway were those of cyclic AMP and IP3. Structurally, both pathways were three-component transduction systems modulated by GTP binding proteins, as I explained in the first video. For historical record, Dr. Farid Murad's group was awarded the Nobel Prize for the discovery of the heme-dependent soluble form of the guanylate cyclase in 1998. His group fiercely denied and even ridiculed the existence of the multi-limbed membrane guanylate cyclase signal transduction signaling pathway. To date, neither the media nor the Nobel committees has rectified their monumental error. Thus, the key question which we asked was, did a one-component transduction system, multi-limbed membrane guanylate cyclase that generates cyclic GMP as its second messenger indeed exists for the polypeptide hormone adrenocorticotropic hormone ACTH? This is shown in figure 1, which shows that ACTH triggers the signal from the membrane guanylate cyclase molecule, generates cyclic GMP, and converts and controls the process of steroidogenesis.
Did you find the answer to the question, is cyclic GMP indeed the second messenger of ACTH?
Yes, we did. It constitutes limb 1 of the membrane guanylate cyclase molecule, yet this limb does not exist in the plant and the insect kingdoms of the universe. Nonetheless, the three kingdoms of plants, insects, and mammals are bonded because they all generate steroid hormones. I will briefly narrate how we solved this mystery. This idea originated during the years 1967 and 1968. At that time, as a steroid training fellow in the Worcester Foundation for Experimental Biology, I collaborated with Dr. Irahu Kaspi. We demonstrated that in the stereochemical pathway of plants and in mammals, and we used models of rats, transformation of mevalonic to cholesterol occurs by almost identical manners. Both generate progesterone hormone. Empowered with this knowledge, in 1969, I independently tested and confirmed the idea in our my wife and mine house in Englewood Cliffs, New Jersey, using tea plant Camellia sinensis. Thereby, the evolutionary concept arose that the nature's three kingdoms, plants, insects, and animals, are bonded, and progesterone is a procreation steroid hormone. This is shown in the figure. Plant, insect kingdoms generate hormone progesterone, and this progesterone is involved in our reproduction. In April 1969, I presented this concept during the annual meeting of the Federation of American Scientists Experimental Biology, FASB, held in Atlantic City. At that time, all affiliated societies of FASB met at the same time and the same place. Such a huge gathering could only be accommodated in the hotels located at Atlantic City's boardwalk. In the presentation, I emphasized this novel common theme as evolutionary concept. That is, that plant, insects, and animal kingdoms are bonded together.
How this novel concept was received by your peers?
The concept received a rousing applause from the conference attendees. Influenced by it, Dr. Abbas Kitabchi, one of the attendees in the conference, introduced himself to me. He had recently accepted the position of Associate Research Staff and Director of Research in Veterans Affairs Hospital in Memphis, Tennessee. He asked me if I would be interested in developing the proposed concept in the VA Hospital Research Division. If I agree, he would like me to direct the steroid research group at VA Hospital, and I will have an affiliated position of associate professor in the Department of Biochemistry in the University of Tennessee Health Sciences. He wanted me to think over this proposal, and he will call me shortly to know my answer.
It was obviously very appealing offer. What was your answer to Dr. Kibachi? Did it take you a long time to make a decision?
Around the first week of May, Dr. Kitabchi called me and invited me to present a seminar on my research in the Department of Biochemistry, University of Tennessee Medical Units in Memphis. I agreed to do so. On the mutually agreed date, in the second week of May 1969. Accordingly, I flew to Memphis, reached there at about noon, was welcomed at the airport by Dr. Kitabchi, and lodged in the famous Peabody Memphis Hotel, located in the central part of the city. The rest of the day I spent with Dr. Kitabchi, who showed me the research division of the VA hospital, introduced me to its key research members, notable among them Doctors Jim Brush, Solomon S. Solomon, Ken Hashimoto, and Edwin Beachy. The visit was followed by meeting Dr. William Byrne, the chairman of the Department of Biochemistry. For dinner, accompanied by Doctors Solomon and Byrne, Dr. Kitabchi entertained me in the restaurant of the Peabody Hotel. The conversation was pleasant, revolved mostly around the research programs of each of the attendees and desirable residency places in the nearby facilities of the medical unit. Next day, I delivered the seminar at 9 a.m. It was well attended by the in-house biochemistry faculty and staff, the veteran administration, and the interdepartmental university faculty. The seminar lasted about 1/2 an hour and included my presentation and also the questions and answers. The seminar was well received. Afterwards, in Dr. Byrne's office, in the presence of Dr. Kitabchi, I was asked if I had any questions about the department and the research environment in the VA hospital. In return, I thanked them for their hospitality and, and commended the first-rate job they were doing in building their departments for academic excellence. At about noon, Dr. Kitabchi drove me to the airport. I flew back to the Newark airport and via a taxi reached home in Englewood Cliffs, New Jersey, where Joan was happily waiting for my return. I discussed Dr. Kitabchi's offer with her. After a few days, she responded that the offer was attractive and Memphis was a desirable place to settle. Consequently, I was eager to have the opportunity to experimentally test my concept and pursue it further if required. I informed to Dr. Kitabchi my decision to accept by phone.
So you decided to again join the academia to further your research. Obviously, a new part of your life was about to begin.
Yes, that's right. About the middle of December, I received a phone call from a realtor from Memphis. He was contacted by Dr. Kitabchi's office to help us relocate to Memphis. He offered his help in buying a house before our move to Memphis. So far, we had not given a thought yet. Instinctively, I answered in the affirmative. I welcomed his help, told him that my wife Joan and I are intending to be in Memphis by the end of December, and requested that he keep an eye on the suitable houses close by to the medical center. On house particulars, keeping in mind that we desire to raise a family, we requested for a 4-bedroom dwelling located in a serene setting with a spacious backyard. By the third week of December, with these attributes, he had identified a few houses in the Bartlett locality. It was conveniently located about 10.5 miles off the northeastern boundary of Memphis. The city limits covered a total area of 23.42 miles. Around the third week of December 1969, Joan and I packed our bare necessity items in our recently bought Buick Skylark car. The remainder were professionally handled by a moving company to be warehoused in Memphis till the purchase of our new house. With sadness, then we left our beautiful house, 81 Grand Avenue, Englewood Cliffs, New Jersey at 6 a.m. and started our voyage to Memphis, covering about 1139 miles via Harrisburg, Pennsylvania, Charleston, South Carolina, making an overnight stop in Louisville, Kentucky. The next day at about 4 p.m., we reached Memphis. Therein, for a few days, we took refuge in a hotel located in the medical center area till the purchase of our future house was finalized. Focusing exclusively on the Bartlett area, we purchased a house addressed 2827 Flowerwood Road, Memphis, Tennessee 38134. Built in 1967, it was only 2 years old, situated in an area of 1,573 square feet plot. It was a 4-bedroom, 2.5 bath property with a two-car garage containing a serene environment. It had spacious backyard. It is pictorially depicted now. Its added feature was that it was positioned in a small town. According to my calculations, given its gross data of about 34%, its population was about 20,000 miles in 1970. It was quite close, only 10 miles from my workplace in the Memphis Medical Units, and notably, it was within 3 miles from the school district located on 5705 Stage Road, the place our future children would start their education. On December 27, 1969, we completed the purchase with a 30-year mortgage. Yet, the original owners requested a delay of 2 days. We moved in on 29th, and in lieu of the delay, the owners offered us a female 6-week-old puppy of pure Shetland sheep breed. We happily accepted the offer, named her Nimki, the sumptuous Indian snack, and incorporated her as a precious member of our family. On December 30, 1969, we occupied and started to furnish the house. The new movers brought in, unpacked, and placed our house belongings in the designated places. To familiarize ourselves with the surroundings, we examined and were satisfied with our fenced backyard and the two-car garage. A pleasant feature of this two-storied house was that its den immediately faced the wooded backyard. Yet it was positioned by a cemented about 400 square feet corridor. We thus frequently and on weekends use the den and the corridor for our evening meals and weekly barbecues.
It looks that on the last day of 1969 you were settled well in Memphis and rank on the new year 1970 ready to start your new professional adventure.
It was exactly this. In the first week of January 1970, I reported Dr. Abbas Kitabchi, the Chief of Staff for Research and Development at the Memphis Veteran Administration Hospital Center. His administrative assistant aided me to cover all the required family formalities for my employment. I was now the director of the Sterile Biochemistry core laboratory. The laboratory was about 600 square feet in area, equipped with standard equipment such as low and high speed centrifuges, incubators, cold room, and normal test tube shakers. There were 2 desks to sit for analyzing and calibrating the experimental data. I was introduced to Mrs. Louise Kitchell, who was presently the lone technician in the laboratory and who would work under my supervision. She was pleasant and welcomed me. I was then shown a 400 square feet office space for my use. An additional about 400 square feet laboratory would be remodeled and would be provided to me. The condition was that I'll be free to test my universal evolutionary concept if I can secure extramural support for the guidance of future graduate or postdoctoral fellows. They will be the pioneers of my envisioned research program, elucidation of the control mechanisms of the hormones at the submolecular level. Here my concept will be the driving force for my future research program. This basic research will be a central component of my academic position in the Department of Biochemistry. Meeting with the departmental chairman, William Byrne, it was decided that additionally I'll be teaching and will be the course director of the steroid biochemistry offered to the medical students of the University of Tennessee. This assignment constituted 10 one-hour lectures per academic year. I was told that the basic financial support to run the steroid biochemistry laboratory in the VA and to reach the biochemistry course in UT will be provided by the respective institutions. Steroid biochemistry was one of the divisional research components of the Veterinary Administration Medical Center. Other were endocrine and metabolism, diabetes, infectious diseases, electron microscopy, and dermatology. They were respectively headed by Doctors Solomon, known as Sol Solomon, James Brush, Adonach Beachy, and Ken Hashimoto. All these divisions in turn were administratively headed by Dr. Kitabchi. He also was actively involved in overlooking the research conducted in the divisions of steroid biochemistry, endocrine and metabolism, and diabetes. While being accustomed to the new culture of academic research, teaching, and waiting for remodeling my office and research laboratory, I spent time in writing and submitting my pending manuscripts, and also bore on preliminary experiment data to find a deeper biochemical relationship between the kingdoms of plants, insects, and vertebrate animals. I published 3 manuscripts on the concept. Having the manuscripts published, I was ready to apply to the basic research department of the VA hospital for funding. I intended to propose the elucidation of the molecular mechanism by which the pituitary gland meets the fasciculator cells and controls its cortisol production, as shown in the figure. The notable property of the model was that it would not be applicable to the prawn kingdom because it does not embody the ACTH limb, and it would provide me the opportunity to enter the new field of endocrinology where unanswered questions remain. I therefore wrote 2 small Veterans Administration Part 1 and Part 2 grant applications to financially support the testing of my ACTH model. Passing through the critical peer review of process, both applications were rated highly meritorious and were funded within 3 months, to my added delight. The Veterans Administration Research Committee also awarded me with 2 commendation certificates accompanying their awards. I have now grown into a significant basic science and clinical investigator in the Veterans Administration Hospital, and the certificates of merit which have been awarded to me are shown. Until then, these studies were being carried out on the shoestring budget. I was alone, had no technical help and financial resources to move them forward. These two Veterans Administration Part 1 and Part 2 grants and their ratings as highly meritorious had now provided me the opportunity to go forward with the model.
At that time, the future looked really bright and promising to you. Did you think at that moment that it is the hour of your life, that the dreams of your boyhood were about to come true?
Yes, I had this feeling. The success was dazzling, and I could clearly remember the overwhelming emotion that I will be able to follow the scientific path imagined in my childhood. I was born on December 15, 1935, at about midnight, with the union of my father, Dr. Anantram Sharma, and my mother, Vidya Sharma. My mother was 16 and a half year old, and my father was about 28 and a half. Their pictures are shown below. I was born in the Jammu and Kashmir Dogra Battalion Hospital, Satwari, Jammu Tawi. Notably, at the time India was a British territory, I recall going to the roof of our house and shouting, "Quit India!" In panic, my mother locked me in the closet because it was a criminal act by the British Empire. In 1945, when accompanying my father in the Dogra regiment in the Himalayan mountains in Bunji Gilgit of Jammu and Kashmir state, situated at an altitude of 8,200 feet above sea level, I crystallized sodium chloride. With the conscious passion set on fire, I started dreaming about my becoming a scientist. On October 5, 1947, at the age of 11, I became fatherless due to the Pakistan Pathan tribal army siege of Punch, Jammu Tawi of the Dogra Battalion, where my father served as a medical doctor, was wounded, video number 1, and later received gallantry award for his bravery. I had to carve my own way. In April 25, May 12, 1953, at the age of 17, I spent approximately 2 weeks' time with Honorable Atal Bihari Vajpayee, future Prime Minister of India. It happened during my summer vacation in the house of the Kishan Kotwal in Bhadrawa, Jammu. Then Mr. Kotwal was the president of Praja Parishad, a political party aligned with Jan Sangh. That later became Bharatiya Janata Party. Honorable Vajpayee was a political fugitive hiding in Mr. Kotwal's house. Then Press Secretary to Dr. Shah Prasad Mukherjee, Mr. Vajpayee, on May 8, 1953, at 6:30 a.m. in a jeep to Jammu, had escaped the police arrest from Mr. Sheikh Abdullah's government. Mr. Mukherjee's team was protesting the existence of the Article 370 applicable to the state of Jammu and Kashmir. The article made the state semi-autonomous, yet prohibiting the residence of any out-of-state Indian subjects.
Obviously, you have had a lot of important memories to be nostalgic about. But back to reality. How did your research progress?
After receiving from the VA Hospital Basic Research Funds amounting to $45,000 a year, I started to build my research group. The long-range task was to decode the biochemical, structural, and physiological principles by which ACTH, secreted from the pituitary gland, controls the excretion of cortisol in the fasciculated cells of the adrenal gland. Slowly yet steadily, I built this research group consisting of 32 fellows. After 54 years established existence of the limbed, multi-limbed membrane guanylate cyclase cellular signal transduction pathway. Without it, the universe will be dark and lifeless. Here is a picture showing the members of my research group. Mrs. Linda Sutcliffe was the first member of my group. She joined as a full-time technician. With the degree of Bachelor in Science from the University of Georgia and also being a member of the cheerleader group of the football team, she was extremely pleasant and eager to learn the new tools of biological research. In the beginning, chiefly, she helped me to do day-to-day hands-on experiments. Then she quickly progressed, learned how to prepare and deal with the isolated cell preparations of the fasciculata and carcinoma cells, how to carry out the incubation experiments and assay the steroid levels. She became an extremely valuable asset. I was very happy to have her assist me. My research advanced significantly, resulted in three major scientific publications. We now had firmly established the model isolated cell lines of the fasciculator cells and the adenocortical carcinoma 494 cells, demonstrated them to be the extraordinary tools of carrying out steroid hormonal research, and thereby shown that the ACTH control of steroidogenesis is indirect, and this control is lost in the cancer cell at the pituitary adrenal axis.
Thank you, Dr. Sharma, for this fascinating story about the early years of your life devoted to science. I am looking forward to listening more about the years which follow the impressive beginning.
In the next video 5, this saga will continue to be narrated, defining the events of turmoil and glory. Thank you.
Thank you.